Can you compare what we've learned from Moderna this morning and contrast that with what we learned from Pfizer last week?
Yes, good morning, John. Can I just first say that this is, I was a little bit, well, I'm kind of speechless to a degree. At this level of efficacy, I was really getting ready for a 70-80%, because there are differences between these vaccines. 94.5%. I'm not going to call this better than Pfizer, because this is, we're dealing with such few cases. But thank God it's higher than that number rather than below it. And we get more from these guys. We're getting more with regards to the statistics. So you see that p-value they put in there, which is pretty powerful, which is not a surprise. We're also hearing that the 11 severe cases so far were all on placebo when you read further down. That's great. And if that carries on being the case, we'll be very happy. And lastly, they do tell us that 15 people in the trial, in the 95 cases, were older adults. But it doesn't tell us which groups they were in, the vaccinated or the placebo. So that's one little thing that we'd love to know.
Sam, the 23-mile difference from Moderna in Cambridge out to Andover in Pfizer's refrigerators is a difference between negative 94 degrees and, as Lisa mentioned, the refrigeration headlines. Are all of a sudden messenger RNA, is it all of a sudden more distributable worldwide simply because of the freezing to refrigeration path?
Yes, Tom. I mean, that's pretty much as simple as it gets. The question for that press release that first came out from Moderna is actually incremental. We knew already that the storage and distribution was at -20. We knew that it was okay to keep it in the fridge at the 2 to 8 degrees for a certain period of time. They've just extended that, and we're going to get more as time goes by about how long these things can be kept. And it just makes the use of the vaccine easier at the end of the line. And, of course, it's already an easier vaccine to distribute than Pfizer.
I mean, this is important, Lisa. Right now we're confusing everyone because I refuse to quote Celsius and Dr. Fazeli refuses to quote Fahrenheit. It's clear, isn't it, Lisa?
Yeah, well, it makes it a lot easier to get the Moderna vaccine out and distributed if you don't have to have some cold ice on the side here. I am wondering whether we can take a 30,000-person study where we're talking about sample sizes of 5 people or 11 people, Sam, and extrapolate that out into something that can really immunize an entire world population. Is this sufficient in your eyes?
Yeah, well, it's a start, and I think this is what I've been saying with regards to the Pfizer already. These are not the levels of effectiveness that we expected at this point. Everyone was psychologically, mentally preparing themselves for 50, 60, 70%, which would have been good. But this is levels that nobody expected. So, again, as I say, we've taken the first step. We've thrown the first set of dice, and we got 2 6s. It's a great, great start.
Sam, just a final question from me, just to wrap things up. Is there a difference in providing this information in a press release compared to a peer-reviewed journal? Can you compare and contrast that? Just that approach and how this information is being distributed into the public.
Yes, the absolute level. I mean, you have to give it out because it takes time to prepare these documents to get them published. You need to know how well the two groups were balanced. If there were lots of people with comorbidities in the placebo group, which therefore didn't respond to the vaccine as well. I doubt that's the case because these are already large numbers, but you do need to see that.